So now I'm going to talk about how viruses escape from effective immune responses. We call it immuno-evasion. So I will show just some examples. For example, hepatitis C virus overcomes the antiviral effect of interferons by blocking protein kinase R. And this is very important for the, as we mentioned, that it is type 1 interferon, the immunity against the viruses. So it has blocking and interference activity with this important protein. And also Herpes virus type 1 and 2 express also proteins that inhibit antigen processing and also inhibit presentation. And as can be seen here, this is now viral infective cell, for example, so the antigens are presented in association with major histocompatibility class 1. So now it will be recognized that this cell is infected by a virus. So some viruses, in order to abolish this or to hide themselves and not to be seen by other immune cells, they tend to inhibit antigen presentation, processing and antigen presentation of viral antigen to CD8 T cells. And adenovirus and cytomegalovirus, for example, reduce expression of class 1 molecules, and actually also this inhibit antigen presentation to CD8 T cells. Here, this is one, for example, and this is CD8 T cell. This is major histocompatibility. So any antigen presented with major histocompatibility class 1 will be seen by CD8 T cells, and it will get actually activated. So also cytomegalovirus, measles virus, and HIV reduce level of class 2 and not class 1. And actually class 2 molecules are very important, as I mentioned in the beginning, that class 2, they play very crucial role as T helper 1 usually recognize antigens in association with major histocompatibility class 2, and so it will produce interleukin number 2, and which is very important, and it has this cytokine paracrine effect on CD8, and it makes it of long life, and it makes it get activated, and it will show its antiviral activity. So cytomegalovirus, they know this information, and also measles virus and HIV, so they reduce level of these molecules of class 2 on the surface of the antigen presenting cells. So now I will show how some viruses evade the effective immune responses, or what is so-called Immuno-Evasion, by inhibition of complement components. So, for example, Vaccinia virus and Herpes simplex virus, and these, they bind to C3b. This is here C3b, C3b and C4b, and this interfere with the complement pathway, interfere with formation of membrane attack complex, interfere with formation of anaphylaxis, which are very important for lysis and important for the inflammation during infection. And however, some viruses may produce immunosuppression, and it will lead to suppression or cause generalized immunosuppression. For example, Epstein-Barr virus or cytomegalovirus and HIV And in case of actually Epstein-Barr virus, they produce cytokine known as IL-10 like. It is not an IL-10, but it is very similar and homologous to IL-10. And as you know that IL-10 has the capacity to inhibit macrophage, and macrophage is very important because they produce pro-inflammatory cytokines like human necrosis factor alpha, interleukin number 1, and also it produces interleukin number 12, which is very important for activation of T helper 1. And activated, as you know, activated T helper 1 will produce in interferon gamma, which has anti viral effect. So the Epstein-Barr virus, they produce this material. It is IL-10 like, and this will suppress the expression or secretion of cytokines by other cells. So also one of the most famous and effective ways for the virus to evade or to escape effective immune responses known as antigenic drift and antigenic shift. And as you know that if we go deep in the virus and to speak about the virus genome, the virus actually, the genome, it is formed of blocks. So these gene blocks, the virus, when it is under attack, may give an order to gene blocks to change their order. So gene number 1 will sit in the place of gene number 7, and block number 7 will come and sit on the block number 1 within the gene locus, and this will produce a new antigen. It is known as antigenic drift. So the immune system is start the defense from the beginning, and when these viruses get recognized by the immune system, so also the virus will give order for an extra process of antigenic drift. But however, this may take 5 years of time. They deal with the immune system and with the environment, and after 5 to 10 years, maybe 12 years in case of corona, so the virus give an order for the genes that make possible target for killing by the immune system, and give an order just to get down regulated and to upregulate very new gene, and this what may lead to outbreak of a disease like what happened in case of corona virus. So also influenza virus, it is follows this way, the antigenic drift and antigenic shift. So antigenic drift, it is actually a matter of deletion, as I mentioned, or insertion or change in the blocks of the gene within the locus. So this actually has led to emerge of new subtypes of, for example, viruses or influenza virus, and this may be known, may occur through genetic reassortment. Assortment means exchange of, in some instances, it is not necessarily for the virus to upregulate new proteins from new genes, but in some instances these viruses may exchange their genes. For example, in the human, may exchange their genes with a similar virus that affect animals, and this is known as genetic reassortment. Thank you very much, Professor Ahmed Boulad.